Welcome back to MLT 240. This is part 3 of the mycology lecture. In this portion of the lecture, we're actually going to talk about some different species of fungi. And in this lecture, we're going to talk about opportunistic fungi. Opportunistic fungi are most frequently isolated in the clinical lab. These fungi fall under the class Zygomycetes. Opportunistic fungi normally do not cause infection in otherwise healthy people, but they do usually cause infections in immunocompromised patients. So our cancer patients, organ transplant patients, neutropenic patients, that's patients with decreased neutrophils, our diabetic patients, and debilitated patients and patients with long-term antibiotics. When determining pathogenicity of opportunistic fungi, symptoms are consistent with a mycotic infection. Fungal structures are present in material obtained from an infected site, and these fungi must be identified correctly, and they're isolated from multiple cultures. Most of our opportunistic fungi are rapid growers. Most form colonies within 4 to 5 days. Most often, these fungi are saprobic, living on decaying organic matter in the soil, and sometimes can become airborne. These fungi are normally inhaled, so they can show up in routine respiratory cultures and may yield few colonies. They may also contaminate the skin as well as other laboratory cultures. Our opportunistic fungi can be further divided into either aseptate or septate fungi. Let's first talk about asept opportunistic fungi. The first species of aseptate opportunistic fungi that we need to discuss is Lipthemia. It is clinically significant because it causes predilation for vascular invasion, can cause thrombosis and necrosis of tissues, usually found in patients with diabetes and ketoacidosis. In this patient population, infection usually begins in the sinuses where conidia are inhaled. Infection spreads rapidly to the orbits or the eyes, face, and the palate, which is the roof of the mouth, and the brain. For cancer patients, other sites of infection are noted. As far as Lithemia for colony morphology, colonies grow rapidly and are woolly. Microscopically, you can see hyphae that are broad and ribbon-like with few septations, erect sporangiospores, solitary or in group, terminate in apophysis, surrounded by a sporangium. Sporangiospores are short and ovoid. Intranodal rhizoids are present. This is an example of what Lithemia corymbifida looks like under the scope. The next species of aseptic opportunistic fungi is Mucor. This is clinically significant because it causes rhinocerebral mucormycosis and disseminated disease. Mucor is commonly isolated in the environment worldwide. It's a rapidly growing fungi at 25 to 30 degrees Celsius. It can form a cottony, dirty white colony that becomes mousy brown to gray with age. For microscopic ID, we have single or branching sporangiospores that support round spore-filled sporangia. Rhizoids are absent, and the mycelium are usually aseptate. Here is our image of our Mucor species. You have the single branch holding up a head that is spore-filled, or sporangia that is spore-filled. Mucor species can cause mucormycosis, rare opportunistic infections of the sinuses and nasal passages, characterized by hyphae growing in and around blood vessels. Symptoms include one-sided facial swelling, headache, nasal and sinus congestion, black lesions on the nasal bridge or upper inside of the mouth that quickly become more severe, and also it can cause fever. The last one in this category is the Rhizopus species. It is clinically significant because it's worldwide distribution and it's recovered from decaying vegetation. It's the most common mycorrhizas causing human disease. presenting as rhinocerebral mucoid mycosis. The colonies for this species are rapid-growing woolly colonies that cover the entire surface of the auger plate. They are initially white, become gray to brown with age. Of course, the hyphae are aseptate. They have erect sporangiospores terminating in dark sporangia and sporangiospores. The rhizoids at the base of the sporangiospores are brown. The sporangia are fragile, do not easily retain when making slide cultures, and can result in umbrella-shaped structures at the end of gonidiospores. Here are images of our Rhizopus species for fungi. For our 3 species of aseptate opportunistic fungi that we've just discussed, there's some differentiating structures that are present, and this slide kind of goes over those differentiating structures. First, there's piriform or teardrop shaped sporangium. That would be like the head, the shape of the head. Leucthemia is the only one that has that structure or that teardrop shaped formation. A well-developed apophysis or swelling at the base of the sporangium. Leucthemia would again be the only species that would have that. Sporangiospores arising between rhizoids and not opposite them. Leucthemia is the only one that does. Mucor does not have rhizoids. And then finally, Leucthemia would have abundant sporulization on routine mycological media. Now we need to cover some species of fungi that are septate and also hyaline, and these are also opportunistic fungi. The first species up is Aspergillus. As far as clinically significant, these are frequently isolated from a number of hospital sites, including vent systems and food. It's the second most isolated fungus after Candida. It's most frequently cause of disease in bone marrow transplant recipients. It causes multi-organ involvement, including the brain, liver, heart, and bone. It can cause chronic pulmonary aspergillosis, especially in patients with structural lung damage due to another disease such as tuberculosis. Aspergillus can cause keratomycosis, which is an eye infection, or otomycosis, which is an infection of the ear canal, and an infection of the nasal sinuses. Superficial Aspergillus infection of the outer ear canal is characterized by inflammation, pruritus, scaling, and severe discomfort. Can result in inflammation, superficial epithelial exfoliation, masses of debris containing hyphae, suppuration, which is a discharge of pus, and also pain. For colony morphology for Aspergillus, we have a rapid grower that produces rugrose, velvety colonies. There is a variety of color possible with Aspergillus, including white, black, yellow, brown, green, gray, pink, beige, and tan. Most pathogens are green to tan. For microscopic observation for Aspergillus, or what we'd see under the scope, we have long conidiospores that connect to hyphae at foot cells. You could see flash-shaped to globose vesicles or rounded shapes for the heads of Aspergillus. You have long, compact chains of conidia arranged in straight columns or radiating patterns. Conidia may be rough or smooth. Here is a macroscopic image of Aspergillus on a media plate. Here's an image of some of the microscopic images you may see on Aspergillus. I want to point out the foot cell, the conidiophore, the vessel, and the chain of conidias. Here's an example of what Aspergillus could actually look like under the microscope. Here's a second example of what Aspergillus could look like under the scope. The next species is Fusarium. For microscopic observation for conidia, macroconidia is going to be more abundant and microconidia is going to be fewer. Macroconidia are banana or canoe shaped and are formed singly in small clusters or clustered together in mats. There's a distinctive soot cell at the bottom of attachment. Fusarium can cause eye infections. Keratitis can refer to a corneal infection that can be caused by either a filamentous fungal, mold, or yeast. Most risk factors are related to some kind of fungal keratitis, including trauma, generally some type of plant material. In the southernmost United States, fungal keratitis cases explain up to 35% of keratitis cases compared to about only 1% in places like New York and Minnesota. This is an example of what fusarium would look like or present on a culture plate. It looks like a woolly or cottony colony. This is fusarium under the microscope. You have a banana or canoe-shaped macroconidia. You can see those here. There's several of them in this image. Next up is Penicillium species. As far as clinically significant, it's inhibited at 37 degrees Celsius, so it rarely causes infection. Most reports of infection involve chronic fungal sinusitis. Penicillium can cause various diseases in our immunocompromised patients. Our colony morphology is a rapid growing colony. It can grow at room temperature on the brain, heart, infusion media. It produces a velvety white colony first, and later it will produce a powdery and blue-green with white periphery. On the underside or the reverse of the plate, it should be colorless. Under the microscope, you will have conidiospores that are erect, sometimes branched. You can have flash-shaped phyllaids supporting chains of round phyloconidia. You can have a paintbrush-like cluster, which is also referred to as penicilli. Here is Penicillium species on a media plate. You've got the powdery blue-green colony with white periphery. Here is what Penicillium species looks like under the microscope. A lot of times it's said to look like a little paintbrush. Tularemycosis. It only affects people who live in or visit certain parts of Asia. The highest incidence is an infection seen in patients with AIDS. It causes bumps on the skin that form that are generally small and painless. Other symptoms include fever, general discomfort, cough, weight loss, swollen lymph nodes, diarrhea, swelling of the liver, spleen, and abdominal pain. While the condition is treatable with antifungals, without treatment, scientists estimate that 3 to 4 people have died with this particular infection. Now we're going to move on to the category of opportunistic fungi that are still septate, but they're diatomaceous. Alternaria is up first in this particular category. It's recovered from almost any source, though primarily implicated in chronic fungal sinusitis. It's found worldwide in grasses and leaves. It's commonly a cause of tomato rot. The colonies are rapid growers. They are light gray in color. They are a woolly colony that matures dark, greenish to black, or even brown, and with black on the reverse side of the plate. For the microscopic exam of Alternaria, the reproductive structure and hyphae are dark. You have short conidiospores bearing conidia in chains that lengthen in a acropoidal fashion. There's multicelled conidia that have angular cross walls and taper towards the distal end. The next species is Orobastidium. Infections are rare, but can be traced to contaminated hospital equipment, may be recovered from blood, tissue, or abscesses. This species is found worldwide, primarily in wet environments. Sources include water lines and shower tiles. For colony morphology, their growth is moderately rapid. colonies have a yeast-like consistency. Initially, it's off-white to pink in color, becoming black and leathery with age. It's a black yeast. For the microscopic Bastidium, the hyaline hyphae create hyaline conidia directly from vegetative hyphae. With age, the dematiaceous hyphae develop and break into arthroconidia, which do not have hyaline conidia. Since this is only a moderate rapid grower, this is what a colony plate can look over a 3-week period. This is week 1, week 2, and week 3. The next species is Cladosporum. This particular species is frequently a cause of disease. It's primarily recovered as a lab contaminant. Infections can typically confine to the sinuses or follow traumatic inoculation. This species is ubiquitous in nature and can be found almost in any location in the world. For colony morphology, growth is moderately rapid, with a granular, velvety to fluffy colony, ranging in color from olive to brown or black, and the reverse side of the plate is also black. What you're going to see under the scope for Cladosporium, the septate hyphae are dark colored. They're short chains of dark 1 to 4 celled blastoconidia with distinct scar at each point of the attachment that are bore from repeatedly forking shield cells. This is just an image pointing out some of the features that we just mentioned on the previous slide for the Cladosporum species. Here is an image of the velvety colony of the Cladosporum species. Here's another example of Cladosporum under the scope. This is the last small section for this lecture, and we're going to cover some other opportunistic mycoses. There's 3 types of fungal sinusitis. There's Mycetoma fungal sinusitis, which produces clumps or spores, which is also referred to as a fungal ball within the sinus cavity. It's most frequently in the maxillary sinuses. The patient usually maintains an effective immune system, but may have experienced trauma or injury to the affected sinus. Then there's allergic fungal sinusitis, or AFS, which is now believed to be an allergic reaction to environmental fungal that is finally dispersed into the air. And the last one is fulminant sinusitis, which usually is seen in immunocompromised patients. The disease leads to progressive destruction of the sinuses and can invade the bony cavities containing the eyeball and the brain. This is an example of a fungal ball, or the implantation of fungus in the sinus cavity. This is usually unilateral. The maxillary sinuses are commonly involved, can be seen in an immunocompetent patient, and the most common cause is the Aspergillus species. This concludes part 3 of this lecture. If you have any questions or need any further clarification, please don't hesitate to reach out through email or during lab time, or come see me during my office hours.